Over the years, scientists have analyzed strands of Beethoven's hair to unlock the secrets of his genetic makeup, with findings related to his ancestry and his unfortunate health. Changing genes was considered impossible until CRISPR, a tool that can be used to modify genes. From designer babies to multi-million medical treatments, CRISPR is changing us one gene at a time. Our DNA contains a vast set of instructions that governs our biological functions. Within DNA, there are specific segments called genes. These genes hold the blueprint for various traits such as eye color, hair type, and many others. CRISPR Cas9, a revolutionary gene editing tool, was inspired by a natural defense mechanism found in bacteria. When bacteria encounter viruses, they employ a two-step process to neutralize the threat. First, the bacterium captures snippets of genetic information from the invading virus and integrates it into its own genome until it's needed. This stored information is then transcribed into a guide RNA molecule, which acts as a molecular GPS to locate the viral DNA. Secondly, a protein called Cas9, which acts like molecular scissors, binds with the RNA. Guided by the RNA, Cas9 precisely cuts the viral DNA, rendering it ineffective. In genetic engineering, scientists have copied this bacterial defense system to edit not only viral DNA, but also other genetic sequences in living organisms. By designing a specific guide RNA that matches the target DNA sequence, CRISPR Cas9 can be directed to cut the desired section of DNA. Once the DNA is cut, the cell's natural repair mechanisms kick in. By supplying a healthy DNA template, the cell can seamlessly integrate it into the cut site, effectively repairing the genetic flaw and thus completing the desired gene editing process. CRISPR stands out as a groundbreaking technology, offering us the ability to edit targeted DNA at specific locations, replace flawed sequences with healthy DNA, including diseases. CRISPR has achieved a significant milestone by developing a treatment for sickle cell disease, which received FDA approval in December 2023. Sickle cell disease stems from a single mutated gene and can cause severe pain, organ damage, and strokes. While this breakthrough is promising, it presents challenges, including its high cost, complexity, and limited accessibility. In the early days of its discovery, Dr. He-Jian Cui, a Chinese scientist, edited the genes of human embryos using the CRISPR-Cas9 technology. The altered embryos were made resistant to the HIV virus. Subsequently, these altered embryos were implanted in women. Dr. Jian Cui faced legal consequences and was imprisoned in 2019. Today, we are moving even closer to similar gene editing practices, but in a safe and lawful manner. CRISPR technology could extend beyond illnesses to potentially changing people's appearances, something resembling a genetic form of plastic surgery. Ethical concerns have risen, particularly regarding accessibility. The high costs associated with treating conditions like sickle cell mean that CRISPR-based treatments may only benefit a privileged few. CRISPR's impact will not be limited to medical advancements. It will revolutionize agriculture, earth conservation efforts, and many other areas. However, the ethical considerations surrounding its use highlight the urgent need for guidelines to ensure the CRISPR technology is employed responsibly, safely, and equitably. Don't forget to like and subscribe to see our next video.